sometimes you get a name called CDC2. Okay? Cyclin B with the CDK1, cyclin C, cyclin D, etc. We'll go to that. So you just get an impression. These are positive regulator of cell cycle. These are the cyclin and CDK. Okay? So if you think about these, it's even much more complicated than the bicycle or than the car system we have. Because bicycle, we only need gas pedal. But for cell cycle machinery to move forward, all these positive regulators have to be in place. And then we will tell you much more about all these regulators. So, following the cycling, here is the CDK. CDK, as I say earlier, cyclin-dependent kinase. Again, has a whole bunch of proteins there. A family, including CDK1, or sometimes you call it CDC2, okay? And each one has its own partner, okay, involving different cell cycle phases progression, including, everybody knows cell cycle phase. There's a G1 phase, S phase, G2, and N phase, okay? Anybody know what's a G1 phase? The G stands for what? Anybody know?
I think the G stands for get.
Good, very good, okay? G stands for get, okay? Meaning that within that period of time, there's not much action going on, okay? So it's a gap for the cell cycle progressions. S, everybody know what the S stands for? Synthesis, yes, indeed. S stands for synthesis, meaning that during those period of time, cell cycle is undergoing DNA synthesis, okay? And N phase stands for mitosis. G, another gap, the gap 2 before the cell move on to mitosis. We'll talk about these cell cycle phases in much more detail. But I just want to tell you, the cell cycle progression, it's not just unique to our human being. As I said earlier, East system has it. C. elegans has it. The worm has it. The Drosophila has it, okay? And then that's meaning that all these cell cycle machinery are evolutionarily conserved. Evolutionary conserved meaning that they are so important for a cell to proliferate. So you get an impression about how these are positive regulators of cell cycle machinery. As we talk about the cell cycle, we talk about positive regulators, which including cyclin CDK, okay? And then we already talk about cell cycle of different phases, the G1, S, G2, and M phase. The M phase is very delicate control because during those periods of time, M phase allow the one original parental cell become two daughter cells. And the chromosome have to be equally distributed into two daughter cells. And so that period of time is very critical. And let's take a look at these different cyclin CDK. What do they do about cell cycle? For example, the early G1 are controlled by cyclin B CDK4 or cyclin B CDK6. Late G1 was controlled by cyclin E CDK2. When the cell goes into the next stage, S phase, was controlled by cyclin A CDK2. The G2M transition was controlled by cyclin A CDC2 or cyclin A CDK1 or cyclin B CDK1. So, to finish your cell cycle, I would like to ask some of you, for a cell, from one cell become two daughter cells, how long does it take? Anybody know? From one cell become two daughter cells, meaning that finish the whole cell cycle. How long does it take? Anybody have any answer? Or just want to have a guess? Okay?
Yeah,
depend on the cell.
Yeah, so it can take from like a 2 hours to 2 days.
12 hours to 2 days.
Yeah, I think so.
Okay, yes. The answer is close, okay? The cell cycle, for mammalian cell cycle. Okay, before I answer your question, bacteria also have cell cycle because they need to finish from one bacteria to become two bacteria. For E. coli, an example, how long does it take for E. coli to from one become two? Anybody have answer? Raise your hand. Yes.
About 20 minutes.
Good. 20 minutes to 30 minutes. Okay. So that's bacteria, really, really fast. For mammalian cell, her answer is very close as well. Roughly about 18 to 24 hours. Okay. 2 days a little bit too long. Okay. About 18 to 24 hours. Depend on cell, as you said. Okay. For a cell to finish the cell cycle. So we have looked at all the cell cycle here. Let me ask you. For mitosis, you could see here, how long does it take for the cell to finish mitosis? Anybody has answer? Want to guess? Just raise your hand. So I got an answer here about 2 hours. Anybody know? 30? Okay. It took about 2 hours is close, but it took about half an hour, 30 minutes to 1 hour. So mitosis finished very quick. Okay. Usually, so that's the reason. Usually, when you culture cell, this morning we talk about cell culturing. You could barely see a cell in the mitosis because they are so fast. Unless you look at them 30 minutes continuous, look at those plates, you barely could see the cell in mitosis. Okay? So you get the feeling about these very interesting regulation about cell cycle. Okay? So we talk about these cell cycle parts regulator. Next one. About these different party regulator for the past 10 or 15 years. The scientists are very interested to know what the biological function of these cyclin CDK proteins. So they have done the so-called knockout study. Okay? Knockout means genetically manipulated for showing to understand the phenotype of the if we lost that particular gene. Particularly this was done in a mouse system. For many of you who have no idea about gene knockout or knockout mice, I encourage you to Google it, okay, because this is a very important tool to understand many genes function, okay. So if you can, Google and try to find out what is the gene knockout, what is the knockout mice, okay? So here, I just want to point to you, these genes are very important for cell cycle proliferation as we find out earlier. But we do want to know how indeed their contribution in cell cycle. So the genetics or scientists try to knock out either cyclin or CDK, okay? Here, just point out to you, the CDK1 is the one we talk about. CDC2 involving M phase. Okay, remember M phase is allow the cell go into mitosis doing the cytokinesis, allowing one parental cell become two daughter cells. CDC2 is very critical because if we knock it out, the embryo died at 1.5 days, which means the CDC2 is so critical. They could not even pass the first fertilized egg embryo to become true cell embryo stage. They die at such an early stage, okay? So you get a sense about the different cyclic CDK, why they are so critical in cell cycle regulation. However, if we take a look, other cyclic CDK, if we knock it out, for example, CDK2. If we knock it out, the CDK2, the knockout mice still survive. They are viable, okay? Except when they grow up, they are now no longer fertile, okay? So meaning that CDK2 may not as important as CDK1 in terms of the developmental process, okay? Also tell us, because we have so many CDK family members, CDK2's biological function, when we knock it out, it could be complemented or replaced by other CDK. Okay, so that's you see here. And then you go on the list, there's a CDK4, CDK6, etc. Okay, seems like they are very viable. Okay, so again, even CDK4 or CDK6 biological function, and if you recall, CDK4, CDK6 is involved in the early G1 stage. Okay? Only CDK1 is involved in mitosis. So if we not get it out of mitosis, cycling CDK, the cell could not go on to separate into two daughter cells. And that's the reason you see it. The gene targeting or gene knockout, the phenotype, they are different. Here it shows you in much more detail. So the scientists want to know indeed how detailed of the contribution of these different cycling CDK. So they not only do single knockout, they do double knockout, or even do the triple knockout. Okay? So now you can see here. Okay? The single knockout of the CDK1, as we talked about earlier, the embryo died at 1.5 days. Okay, this is E meaning embryonic state. That means when the embryo just formed after fertilization, the embryo starts dividing into, proliferating into the more cells. And then after the fertilization, right away, that E point 1.5 days. If the E 12.5 days, meaning that the embryo developed after the fertilization.